Yes, today we will study the cell diagram of an electrochemical cell. The following conventions or notations are applied for writing the cell diagram in accordance with the IUPAC nomenclature. Recommendations that the cell is represented as this is the representation of zinc copper cell. Okay, so the anode is zinc solid is converted into Zn2 positive and the concentration is C1 and that is written in the brackets. And then double line is there, then copper C2 concentration and copper solid. So first point, anode half cell is written on the left hand side while the cathode half cell on the right hand side. So when we write cell, you have to first write the anode, then afterward cathode. A single vertical line separates the metal from aqueous solution of its own. Like the zinc solid is converted into zinc ion, the single line is there, vertical line is there. Now the third point is a double vertical line represents salt wedge which allows the passage of ions through it but prevents the mixing of two solutions. The fourth point is the molar concentration C is placed in brackets after the formula of the corresponding ion. Fifth point, the value of EMF of the cell is written on the extreme right of the cell. For example, the cell and I have written is downward, that is EMF of the cell is plus 1.1 volt. Now the next point, sixth point is in an inert electrode like platinum is involved in the construction of the cell, it may be written along with the working electrode in brackets. Say for example, when a zinc electrode is connected to a hydrogen electrode, so we write zinc solid, zinc ion and then H plus C2 concentration and H2PT. So this is the representation of the electrochemical cell. In the electrochemical cell, the reactions are reversible. So the reactions occur due to the difference between the electrode potential of the two electrodes. So there is the reversibility in the electrochemical cell because the ions as present in the solution and metal rod is common. So if the potential difference between the two electrodes is equal, then there is no flow of current between the electrodes. Now the first point, when the external voltage is less than 1.10 volt, the electrons flow from zinc to copper, but current flows from copper to zinc, that is in opposite direction. Zinc dissolves at anode and copper deposits at cathode. You can see in the diagram, the first diagram, the current is flowing and the copper is depositing on the copper electrode. So the potential difference between the two electrodes is less than 1.1, outside is 1.1. When the external voltage applied is less than 1.1 volt, then is increased slowly. It is observed that the reaction continues to take place till the external voltage attains the value 1.1 volt. When this is so, reaction stops altogether and no current flows. When the potential difference will be equal inside outside, then there is no flow of the current in the cell. Now third point is the value of external voltage exceeds, if we are applying the voltage that is more than the 1.1 volt, because the reactions are reversible, so cell will be reversed. The reaction takes place opposite direction. The cell functions like an electrolytic cell or the reaction is reversed. So these cells are working on the basis of the potential difference between the two electrodes. And when we apply the external electrode potential, then the reactions may be reversed because the reactions are reversible in the electrochemical cell.